Good morning.
How are you?
I'm okay. When are your students speaking?
Next week.
Okay, oh good. You know, I did not look again at the schedule to see if we added anyone. I remember there were 4 presentations listed a week or so ago. Is that still the same?
Oh, I'm going off the website, but I can go
Okay.
to the spreadsheet real quick.
I'll check. No, I'll check. I should have done that earlier. Looks like we've got, yep, 4.
Okay.
Yep, good. Yeah, when I set up the schedule on the website, I just copied everything from the Google Sheet, and it doesn't load the live version of it.
Yeah. I've got my cat again.
This is the people-friendly cat. What's your cat's name?
This is Echo.
Echo. All right.
He's very talkative.
So does that mean that the cat responds?
Yeah, I mean, yeah. I have a video of myself singing to him, and he, like, meows on cue.
Wow. This sounds like
It's a little weird. I'm weird.
no, this sounds like a contest entry. Yeah. Way back in the old days, David Letterman used to have people do stupid pet tricks. Yeah. Those bookshelves are pretty full behind you.
Yeah, it's my husband's books mostly. I'm an ebook person, so like
Okay.
a lot of the stuff that I read is digital. But
Online.
yeah, he likes books. He likes the real books. He's one of those people who doesn't like the e-readers.
It's the tactile sensation. Yeah.
Yeah.
There
And
is something to that. Yeah.
yeah, I mean it doesn't work for all books. I found that textbooks are unruly in the digital format for me, just because you can't like flip to the spot where you know something is, you know?
Right. And so many of them, the pages are so large that you can't ever see the full page on screen at the same time in a font that's large enough to read. So
Yeah. Yeah.
Hey, everybody.
hello.
I think I now know how to assign people to breakout rooms, and I will write something down with enough detail so that I can understand it and send it to you. And I would have never gotten it without a little help from IT. So,
Sounds good. Did you get the recording of?
you know what? I have not. So let me see if I can track that down.
If you log into Zoom into your account, there's a tab for my recordings.
Okay. All right.
I think that's where it should be.
Okay. I will work on that while we're getting started.
Sounds good.
Seems like we usually have a few more. Should I wait a minute for people
Yeah,
to
I think
sign on?
so. Yeah, wait just a minute or two.
Okay.
I have 3 that should be on their way. Maybe they're already there. Yeah, I've got 3 that should be coming.
Okay. Well, it looks like there's a group called.
Oh, Dr. Goins research. All right. Yeah. So
Yeah. So it's probably a few. Maybe people are logged on together.
Yeah.
Right.
Okay, I am so impatient about starting slightly late. So I'm gonna go ahead and get going with announcements. I'll repeat them at the end as well. Welcome to our brown bag session for today. Couple announcements before we get going. Just to remind you that the deadline for abstracts is on Monday, the 13th. The deadline for poster uploads is July 20th, so another week after that. And you'll get an email with instructions for both of these uploads from the Research Matters email account in the next few days, or on Thursday, so tomorrow. So just a couple things in terms of the presentations today. So we really want to encourage you to participate with questions, students, and we've noticed we haven't had as many as we might like, but I just want to remind you that you can ask questions that aren't directly related to the content of the research. So you can ask things like, what kind of equipment did you use? What's the process been like? What was something surprising? Things that you could ask to anybody, but you could get interesting answers from your fellow student researchers. So just keep that in mind, and remember that you can type questions into the chat window or speak up aloud. So we really appreciate your questions. All right, so I think that's it for announcements for now. It looks like we have a couple more participants. And we can go ahead and get started with our presentations for today. The first presentation is from Maggie and Laura, who are talking about modeling close binary central stars of planetary nebulae.
Thank you. Maggie's going to pull up the presentation.
Yep, I'm getting there.
All right, cool.
Oh boy, we are all the way at the end. Hold on. Okay. Thank you for the introduction. As you said, we're talking about binary star systems and planetary nebulae, and specifically, we are talking about the modeling of these systems. So I think the first question here is, what exactly are planetary nebulae? and they are part of the end of a star's life cycle. And they come from stars similar to our sun that are about 0.8 to 8 times its mass. So when the hydrogen core of these stars runs out, the outer layers start to expand as the core contracts, and the star turns into a red giant first. Then this process continues, and the outer layers continue to expand, and they eventually get ionized and glow, which is what you see in the colorful part of the nebula. And the core becomes a white dwarf, which is just a very dense, hot star. So for a couple examples of the shaping, because as you can see, they have a lot of variety in their shapes. So the Ring Nebula on the left looks pretty round, but it's actually in its 3D. In 3D, it's actually like a barrel kind of that we're looking down into. And then the Twin Jet Nebula on the right has like an hourglass shape to it, and it has a couple different layers there as well. And then here's two more just as examples of the variety in these shapes. And oftentimes in 3 dimensions, they have very complex shapes as well, even more than we can see here. So what causes these shapes is one of the biggest questions that we have about them, since they vary so much. And one hypothesis is that binary systems have a strong influence on the shaping of the nebula. So on this plot on the left, it shows binary inclination and nebular inclination, and then it has a couple planetary nebulae plotted on there. So the likelihood of all of these aligning on this slope randomly is just about 1 in a million, but if binaries do influence the shaping strongly, then the likelihood that they will align like this is pretty high. So that's good evidence to binary systems having a strong influence on the shaping. One part of this system that we have to talk about for, one part of this process that we have to talk about for binary systems is the common envelope phase. And that is where one star swallows the other and it pulls them into a closer orbit or even a merger. So in this image on the right, the colors represent density, with purple being higher density and red being lower density. And then the top image in all the pairs is a top-down view of the system, and the lower one is a side view. So you can see at the top that the smaller star is beginning to gather mass from the larger star. And then in the lower images, they begin to spiral together until they eventually merge. And as I said, they don't all merge, but it will bring them into a closer orbit at least. So most, if not all, objects that are close binaries with a white dwarf must come from this common envelope stage. And it releases a lot of energy that can go into the outflow of the atmosphere to create the shaping. So what does the system look like after this? It can give us a couple different results. It can give us type 1a supernovae, which is just a white dwarf that reaches its mass limit and explodes. It can give us a cataclysmic binary system, which is a white dwarf that's gathering matter from its companion star, and there's explosions all over its surface. And we can also do some studying of gravitational waves, particularly in systems that have 2 white dwarfs because they're so dense. But in order to get here, we have to find out some properties of these binary systems themselves. And at this point, there's about 70 known close binaries, but fewer than 20 of them are actually modeled. And so it's important that we can go and find more models for these systems and get values and ranges for their masses, their radii, temperatures, the inclination of the system, and the separation of the stars. So we're working on that with a couple systems this summer.
In order to find all the parameters for our binary systems, we used a program called Binary Maker 3. This program allows us to take the information we observe from the star system and simulate different masses, radii, temperatures, and inclinations until we get a curve that matches our data. The data we look at is how the light changes over time and the radial velocities of the stars. I will use these GIFs to explain exactly what that means and how this information helps us. The left GIF is a light curve plot, which shows the variations in light over one orbital period. The intensity of the light, or flux, is along the y-axis, and time is along the x-axis. If both stars are visible, then you can see all of the light. But when light is being blocked, such as during an eclipse, you get dips in the graph. These eclipses are very important because they help us determine important information such as radii and inclination. The GIF on the right represents a radial velocity plot. The y-axis shows the velocity of the stars towards and away from us, and the x-axis still shows the orbital phase. We can see these velocities in the spectra as a Doppler shift. This image only shows one star's curve, but using both stars' velocity curves together, we can determine important information like the masses of the 2 stars. Slide. Our goal is to get our blue models to match the red data. There are lots of parameters that affect the shape of the curve, so I'm going to take a moment to briefly discuss one. In this example, the difference in temperature between the two stars is greater than in the data. I can tell this because the light curve on the left has one eclipse that's too deep and the other that's too shallow. The other parameters also have specific ways they impact the light and velocities of a binary system. So by understanding how these parameters affect our models, we can compare our models to the data and figure out what we need to change in order to get a better fit. As a quick note, these plots are just screenshots of what the software generates, so they're not very polished. And when I finish my work with the system I'm modeling, I'll be regenerating them in a cleaner format. Slide. There's also an important effect called irradiation. This happens when a hotter star is close to a cooler star, causing one face of the cool star to heat up, as shown in the graphic by Professor Hilwig. This results in a more curvy appearance to the light curve as different parts of the surface of the heated star become visible while it rotates. Slide.
So in order to familiarize ourselves with Binary Maker 3 before getting into some of the actual data we've been working on, we did a practice run with the system HATR7, which has published values, unlike the systems we're working on now. And then we could have something to compare it to see how our work turned out. And so in this graph on the right, you can see that there are 3 different filters of light, and they're the small blue dots, the green dots, and the red dots. And then our model is the light blue curve there. And ideally, we want to find something that when you just adjust the filter, that it'll change to fit all those different, it'll fit all those different curves.
So looking at this curve, we can see a few patterns. First off, that there's no eclipse, which limits some parameters. And there's also the irradiation effect as seen by the curvy appearance. And so once we know kind of the general shape and kind of what the parameters should be for that, we use our knowledge of the different parameters to get a more precise model to make the light blue dots, our model, match all the filters as well as possible. And finally, in order to find all possible solutions, instead of just one possible solution, we adjusted each parameter to the maximum and minimum value that could still fit the curve. And this is how we got the ranges you see here. Slide. Here's an image of the system I've been working on, LO16. The big oval just shows that the outside is much fainter than the inside, so the 2 parts of this image have different contrasts. Slide. The top plot is the light curve in 3 color filters like HATR7. And looking at the system, I see a small eclipse and an overall curvy shape that indicates the irradiation effect. And the radial velocity curve is especially interesting for this system because it's not just a normal sine curve, and I had to experiment with many different parameters in order to get that bump you see in the black curve. And fitting the light curve at the same time is also often a challenge because if you change one parameter, a lot of other things will change and then it won't fit. So once I got one solution, now I can work on fitting, getting as many different values as possible in order to get my full range. Slide.
And this is the system that I was working on, that I am working on this summer. This is not a great picture, but it's unfortunately just the best one we have. The central star is indicated there around the middle with the 2 purple lines next to it. So I only got this data at the end of last week, so I don't have a ton for it yet. But one thing that has stood out to me about it so far is that it only has 1 star on the radial velocity plot. You can see that on the right, all the little, there's not many of them, but the blue dots that follow along with that smaller black curve, that is the companion star. We do not have one for the hot central star in this system. And unfortunately, we will not be able to get that data either. And so that limits our ability to see the masses of the stars very well. So my current approach on this one is to vary the mass ratio of the stars and get different fits at different mass ratios so that we have plenty of options to cover for that missing data. One difficult thing is that sometimes you can find a fit, but it doesn't physically make sense. So like the one I have pictured here, it looks like it fits okay, but when I checked the values that it produced, it gave stars that had a mass of 2 solar masses. And normally in these systems, we can't have anything over about 1.4 solar masses. I have found a couple other physical matches, but there's not really anything consistent for a range yet because I'm still working on it.
So moving forward, Maggie and I will continue working on modeling our systems. We both have a possible solution, but this is only a small fraction of the total work. We will continue exploring the full range of parameters like we did for HATR7. This is the lengthy process that must be done manually, but it's very important because of the 70 known closed binaries, less than 20 have had this work done. This information will help us in our efforts to get a more complete picture of how binary star systems affect stellar evolution.
And here are our acknowledgements, and we'd particularly like to thank the Indiana Space Grant Consortium for the funding for this project. Thank you.
Thank you.
Thank you, Maggie and Laura. You've already got a question in the chat window from Paige. It says, what are some factors in space that could cause interference in collecting your results? Are there things out there that could warp the readings?
That's a really interesting question, actually. So in terms of our modeling specifically, we just received the data from, like, Professor Hilwig got the data from his contacts, and we're just generating the models. So it won't directly affect the modeling so much, but in gathering the data itself, there are definitely things that could affect it, like, for example, the inclination that we had mentioned. I'm sorry, my webcam doesn't work. Gestures could help explain this, but the inclination, if the system is like side towards you, so you see the two stars, you're going to have a lot stronger eclipses. But if it's tilted, so you're kind of like looking down at them and they're going in a circle, then you're going to have less of that, and you can actually determine a lot less information about the system there. So like that's one thing, is the orientation of it in relation to us. And another thing, like for example with mine, I had mentioned the radial velocity plot only has one star on it, and that is because, I don't know the full details of it, but Professor Hilwig did explain briefly that something about the, let me see if I can go back to one of the slides. Wait, I went past it. Yeah, so Laura had mentioned the spectrum here, which is where you get the radial velocity from, and something about my system, we cannot view the spectrum for the hot central star, which is, I wish I could answer your question more specifically, but I don't remember exactly what he said is causing that, but it is some kind of interference with the system that is preventing us from getting that information, unless we could access a larger telescope, but that's kind of difficult to do.
I can add that real quick if you'd like.
Oh, go ahead, you first.
So in order to be able to measure these Doppler shifts, you have to be able to see a line in the spectrum, and sometimes these hot stars just don't show any lines, and that appears to be the case for this system. So without any lines in the spectrum, we just can't measure what its velocity is at any time.
I was going to mention also, we know that there are obviously uncertainties, but the way that we are analyzing this data as well in the program we're using, Binary Maker, it doesn't actually display those uncertainties either. And we're mostly just kind of trying to fit the curve, not by like any specific statistical method.
Good answer. That clarified some things for me. The next question is from JD, who asks, how does Binary Maker work? Do you have to write code within it?
I can take that question. Maggie, can you go back to the temperature slide, just so I have like a picture up? So basically there's 4 windows in Binary Maker. There's the 2 bottom ones you see there that they show like the data, which are the red points and on the left and the blue points on the right, as well as your model. And basically you start with just the red points, like on the left, or the little blue curve on the right. And you look at that and you like kind of look for the patterns, and you're like, okay, I think that like X, Y, and Z are probably part of this system, or things that have an impact on it. And then you go into one of the other windows and you enter in parameters, and it will like let you enter in things like the mass ratio, temperature, inclination, as well as a whole bunch of other stuff.
It looks like
oh yeah, you can pull it up. Yeah, don't you have a window up right now?
Yeah, so
yeah, so that's
this
like
is what the entering looks like. Just put in the numbers and it'll pop out the other.
Can you pull up the
windows.
little, like the part that rotates as well? There's another window. That's the other one. So now you can see all 4 windows there. It will also kind of show the system as it goes around. Could you even just like run it once, just so they?
Sure. Yeah. This is HITR7 because I had it back up to check something, but
okay.
This is what it looks like.
And it kind of generates your model going around. And then if you're like too high or too low or too narrow, you kind of like figure out what you need to adjust in order to make it fit better.
I personally think watching the stars go around is the most fun part.
Yeah, it is fun.
Excellent. Our next question is from Dr. Goins' research students who ask, what are some of the future applications of your research?
So we kind of touched on that briefly about like talking about the type 1a supernovae and the cataclysmic binaries and gravitational waves. This is something that we really want to know how these systems are produced. So we have to model and study the binaries themselves that precede these systems. And type 1a supernovae probably have the best example of that because they're used as a standard candle in astrophysics, which is something because they have a consistent luminosity that like the same brightness, and so they're very easy to use to measure distances in space. So if it looks like, I think it's like if it looks dimmer, it's probably further away or something like that. And so those are very useful for discovering information about a number of other things as well. And then gravitational waves is obviously a very new thing that's being studied now, and so getting information about these for that as well.
Great. So I think, I don't know, we have several more questions. And I want to get to all of them, but I also don't want to run over too much more time. So maybe we can do like one or two more questions. So the next one is from Professor Zygmunt, who asked, it looked like Maggie and Laura independently tried to model one data set. When you did that, did you already know what the published results were, or did you start without that knowledge?
We started without that knowledge. The goal was to kind of see how close we could get on our own.
Yeah, I don't think we even like really conversed about it until much later after Laura had already finished HATR7 and I was just getting to the end of it when we talked a little bit about it. So our values, like they're not exact, but they overlap and they're similar. So that showed that like we could probably move on to do more data. And this is something too that we could probably compare to other published data. So it's not just two people doing this and coming up with the values. You know, there's probably a lot of other people that have published options for this as well.
Yeah, to be clear, my results, which were the published values, actually used additional constraints that I didn't give to Maggie and Laura because I didn't want to limit their kind of modeling in those ways. So we didn't expect theirs to be exactly the same as the published values because they didn't have a couple of those constraints.
Great, thank you. And then last question, is there this is related to a previous question about the spectrum lines. Is there any particular reason as to why the spectrum lines don't show up for the hotter stars?
Yeah, I'm not sure if this is the exact right effect, but I remember reading about basically if one star is like significantly way brighter than the other, you can kind of like swamp out the other star. And so if there's just like a big difference between them, the one might just not be bright enough for you to see it compared to the giant fireball right next to it, basically.
Yeah, that's one reason. The other reason would be radiative transfer physics and basically the line formation and what densities and temperatures those lines can form at, and what the contrast is essentially between the line depth and the continuum. So sometimes the physical conditions of the atmosphere are just wrong for those lines being able to form in the visible part of the spectrum.
Great. Well, thank you for all your questions, and good job on your presentation, Laura and Maggie. I think for the sake of time, we should move on to our next presentation, which will be given by Wenxuan and Crystal about updating the data acquisition system for the diesel engine test cell.
My screen sharing is gone now, right?
It is.
Okay.
Hello, can everyone hear us okay?
Yes, I can hear you.
Okay, I will share my screen. Okay. Okay, Hello, everyone. My name is Crystal. I am a senior electrical engineering major.
Hello, I'm Wenxuan Meng. I'm the senior student of mechanical engineering.
And we are updating the data acquisition system for the diesel test engine test cell. And our professor is Pete Johnson from the College of Engineering in the mechanical department.
In this presentation, we will talk 6 parts of our project. At the beginning is project overview, and in the first week, what we learned, the sub-test we did, and technical issue, our planning in the future, and conclusion. For the project overview, we want to replace current AVL, the system, to NI equipment. This one is the AVL old one, and this thing is the monitor system shown in the computer. In the right side is the engine, which we are working for, and you can see some like gray wells, white wells connected to some places in the engine where we need to test temperature or pressure signals. About the reason why we want to replace it, the AVL system, since it's outdated hardware, and once we want to upgrade, it's overly expensive. And we want more temperature and digital inputs of it, but there is no places to add it. And the last one reason is the internet sensitivity. So for the data mean targets, we want the temperature signals with K and J tap thermocouple, and pressure transducer signals, also the digital inputs. In the first week, we learned the NI compact rail hardware. You can see that in that picture. And the compact rail is the main controller and is connected to the Ethernet, which allows us to program and monitor on any computer. Each compact rail has modules. You can see that in that picture. They are connected together, and each module is specialized to certain inputs or outputs. For example, that is the temperature module. The third thing we learned is the LabVIEW programming. And the LabVIEW is a version programming software. And also we learned the FPGA programming. The FPGA programming has different procedure than the standard LabVIEW programming. Once we want the impulse which changes too fast, the FPGA one is need to be used. And the third thing we learned is the basic machine of the engine, and this is an example of the parts of the engine.
So our first test that we did was testing the temperature thermocouples that are being used in the lab. We have 2 different types, a J-type and a K-type. The difference between the 2 is the metal material that they are made out of. And each metal material consists of different temperature ranges. But since we only have J-type available to us right now, that's what we were testing and calibrating. So we just did a simple experiment where we use this water heater here, and we insert the thermocouple probe inside along with the thermometer. And we compared the results here shown on the graph. And we had a pretty good result. We had a 1.3% error, meaning that this thermocouple was successful enough to use for the engine. And then the next test that we did is our pressure test. So the engine consists of many pressure transducers, and what a pressure transducer does, it converts the inputted pressure into a current or a voltage. In this case, we are working with current pressure transducers, and these are some of the ranges of the pressure range and the current range for this particular one. Some of the limitations, however, was that the module that we had available to us for the summer is a voltage module. So we needed to do some conversion of current to a voltage by adding a simple resistance to it. And that's how we were able to collect the data in our program. And for this test, we did 2 main tests. Test 1 is to test the pressure at a given voltage. Because we needed to do some calculations as to what voltage would match to what pressure, this test kind of verifies our math And then the second test is more looking at the pressure. What is the max pressure that this pressure transducer can hold, and what would be the voltage and current in that given pressure? And we also had some limitations to this test, but we'll discuss that later on. And finally, our last test that we just finished recently is our digital test. We use this anemometer, which collects wind speeds and it creates a digital signal. We use this anemometer. It's not part of the engine. However, we used it as a convenience to get a comparable signal for the period per revolution. This is the flow meter that's actually connected to the test engine. However, it's a little harder to access, so we wanted to limit the amount of usage for that. So we used the anemometer to confirm our code. And once we did, we followed the circuit connection, and you kind of see more of a visual, like real life type of connection here. We had these are the modules that Wen-Shan was talking about, and we had them connected to the oscilloscope that gave us the waveforms and confirmed our program. And this is the LabVIEW program that we were mentioning. It's a lot more visual. So the top part is calculating the digital input and receiving a period out of it. So this is the main input that goes inside a digital logic configuration here. And basically, it takes in the result, the ANDed version of the current signal and the previous signal. And if both signals are to be true, it enters this case structure, which kind of acts like an if-then type of statement, where if the signal is true, meaning it's on the rise clock signal, it will collect the current timestamp and the previous timestamp and gets the difference between the two in order to get the period. The period for the LabVIEW program is collected in ticks. One tick is equal to 25 nanoseconds. So we just did that conversion to seconds, so it's a little easier for us to read. And we do this main cycle over and over. And then we use this other program down here. This is kind of more like the easier, friendly version of the main program, where this icon here references this main code on top, and we get to select what variable we want to read and record real time. So in this case, we want to record the seconds or the period, and then that's when we can do the calculations for the frequency along with the flow rate, which is gallons per minute. Some of the technical issues that we ran into this summer, first was just LabVIEW design in general, trying to learn a little bit more about the background. We had some compilation issues, and one of the, I guess, downsides to LabVIEW design is when you use FPGA programming, there's very long upload time. You know, if we're lucky, upload time can be 5 minutes, but sometimes it can go up to 15 minutes. So it kind of slowed us down a little bit when it came to programming. For the current pressure transducer, there's very little online documentation on the particular ones that we use. We had to contact AVL directly for their documentation. And this was the documentation that we were given initially, which was a little hard to decipher. It took a lot of trial and error to figure out how to get the actual calculations out from the pressure transducer. And then finally, the engine water pressure also proved to be a challenge for us because the flow meter is connected to a larger pump system in the engine. So when we did the first test, there's so much water pressure being caused inside the pipes that in this particular part of the engine, I don't know if you guys can see there's zip ties connected here. This actually popped off and there's coolant fluid inside. So if you have too much pressure, you know, a lot of things can come off or we see a lot of leakage in the engine. So those were some of the issues that we were facing.
So our planning ahead is this one is we designed with SolidWorks a new box to replace the AVL old one. The main equipment of the new box is the NI compact reel we talked about before, and the power supply and the DC current converter. And the second one is we will complete a master lab view program, and we will test and verify it with a full running engine. In a conclusion, we finished pressure, temperature, and digital tests, and we are designing a new box to replace AVL1, and we will add many wells of it, and we will complete the lab view program for the engine.
Any questions?
Excellent presentation. In the chat, Paige asks, what are some benefits you get from AVL that you don't see in the LabVIEW program?
That's a good question. There is a lot more benefits to AVL currently, just because it is a pre-programmed software from a big company. Some of those include like more safety features. That's something that we plan to do, hopefully if we have enough time in the summer. It's a lot more safety implementation. There's a lot more alert systems, a lot more, I believe there's more sensors to it, but that's because it's more sophisticated programming for this device. But like we mentioned earlier, the upgrade is just so costly that, you know, our professor figured that this would be a good opportunity to just have students be more involved in this process and just create a program that we're used to and we're comfortable with, and kind of make that switch. And I believe we have a question. What does AVL stand for? Honestly, I searched it up too, and it is I believe, Austrian company. So the name is very long, and I won't be able to pronounce it. Sorry. So it's just it's the Austrian name of the company. I don't know exactly what it stands for in English, at least. Any other questions? Go.
Thanks, Professor Zygmunt.
That's why I cannot pronounce that. If someone can do that.
We should ask Professor Venstrom, right? He was in Germany.
Right. Anstalt für Verbrennungskraftmaschinenlist.
There you go.
Thank you. What was sort of the most unexpected thing that's happened this summer for you?
Most unexpected thing, I would say when we were testing the digital signal, the flow meter, when the zip type parts popped off, that was quite the surprise. Our professor had warned us before that it might happen. There's 3 main pump components to the engine, and each pump has a different cylinder size to it. So the flow meter has about, I believe, 3/4 inch diameter type of piping to it, and the other ones have bigger ones. And there's valves that we can open and close in order to move, control the water flow, basically. And our professor had given us a setup that we thought, we should be okay with this setup. But as soon as we turned it on, we hear a loud pop noise, and all of a sudden fluid started coming out. So that was a little unexpected, but now we know that, don't just turn on the water system right away, because it's very high pressure. So it's just learning how to close the valves and open things slowly and gradually. I would say that was the biggest shock. It was fun. I mean, it was a good good experience.
Great. Thank you so much. I always hate it when I hear pops in the energy systems lab. It's just not a good thing.
Yeah, no.
Thank you so much for your presentation. And
you.
I think if there are no more questions, we'll move on.
Okay.
All right. And the next presentation is from Alan and Julie, who will be talking about battery application of silver molybdate structures.
Hi. Can you see the screen?
Yes.
Cool. There we go. Hi. So I'm Alan.
I'm Julie.
And today we're going to talk about battery applications of silver molybdate structures. So just to give you a brief outline of what we're going to talk about, we're going to give you some background information about batteries. Then we're going to talk about our battery, the materials that we use, how we build them, and we'll show you some of the results that we've gotten. And then we'll talk about future work acknowledgements and references.
All right, so a little bit about the background of the battery. So what exactly is a battery? A battery is a cell or a container that consists of an anode and a cathode that are connected by some kind of separator. To explain the concept of a battery, we're going to look at the implantable cardiac defibrillator, or the ICD battery. So in this battery, we have our cathode, which is right here, and it's made up of silver ions and vanadium oxide. And then we have our anode right here, which is a lithium metal. So when the battery operates, electrons are transferred from lithium metal to silver ions. Those silver ions are reduced to silver metal, which are these guys right here, and displaced by the oxidized lithium ions. So there are three things we are going to focus on when looking at a good battery like this. The first factor is capacity. So capacity tells us how many electrons we can hold in our battery. The bigger the capacity, the better the battery. So to improve the capacity of the ICD battery, we would want more vanadium. The more vanadium we have, the larger the capacity is. Next, the next factor is energy. So energy tells how much power is harvestable. So in the case of this battery, we would want more silver ions. Silver ions reducing to silver metal is the reaction that gives us more power for the battery. Finally, the last factor is speed. Speed tells us how fast our battery can perform. The faster the battery, the faster it can move its electrons, which means it can discharge and recharge more quickly. So in the case of the ICD battery, we would want to generate more silver metal. Silver metal is a highly conductive material, which allows it to move its electrons quicker. So after that spiel, if you're going to take away one message from this presentation, it's that we want batteries with larger capacities. So the figure on the right-hand side right here, both the anode and cathode are larger when compared to the figure on the left-hand side. The figure on the left-hand side has a battery that can only power something such as like the 2003 Nokia cell phone. So we don't want that. Now the figure on the right-hand side can power something such as the new iPhone. So we like that. Now, even though we want batteries with larger capacities, there are always other factors that we need to consider. So for example, we question and we ask ourselves, how much material do we use? How safe is this material? How much does it actually cost? How much energy can we get out of it? And finally, is our battery rechargeable? So referring back to the ICD battery, instead of using vanadium and lithium, we would want to use something such as molybdenum and zinc.
Yeah, so now I'm going to talk about our actual battery material. So as Julie said, we want to replace lithium with zinc. So we want to use zinc because it's a relatively cheap material. It's typically not rechargeable and it has a short battery life, but it's really good at discharging energy. And the other good thing is that zinc is a lot less hazardous than lithium. Lithium's flammable, so we could potentially explode when using it. I'll talk about molybdenum in a few slides. So now I'm going to give you a general idea of how our battery should actually work. So here we have a zinc anode. This will donate 2 electrons and turn into a zinc ion. These electrons will be picked up by silver ions that are present in the battery material. Once silver picks up electrons, it turns into silver metal, and it creates a gap where zinc ions will swoop on in and fill it. This process continues to happen where eventually we've replaced all of our silver ions with zinc ions and we've created silver metal. So now I'm going to tell you about the actual material that we're going to use. So these images that I'm showing you, these are the atomic structures of what our material should look like. Here, blue represents molybdenum, silver represents silver, and red represents oxygens. So these first two materials that we have are known as our monomolybdate sample because there's one molybdenum, and our trimolybdate sample because there's 3 molybdenums. In these pictures, you see a lot more than 1 molybdenum and 3 molybdenums, and it's because this just represents the simplest chemical formula that we have. And between these two materials, they obviously have varying ratios of silver and molybdenum. Our next two materials are known as our monoclinic sample and our triclinic sample. As you can tell, they have the same chemical formula, but they have completely different atomic structures. And we're hoping with these 4 materials, we can answer 2 questions. How much silver actually matters? So with these first 2 materials, we can see if we want bigger ratios of silver or if we want bigger ratios of molybdenum. And the second question we want to ask is, does the location of the silver actually matter? So this will let us know if we want materials that have more silver on the surface versus materials that have silver that are that's spread out throughout the material. And just to give you an idea of the actual chemical reactions that are happening, we will reduce 2 silver ions in all of our materials, and we're choosing molybdenum instead of vanadium because with molybdenum we have the possibility to reduce it to a +4 state, whereas vanadium, typically you can only reduce it from a +5 state to a +4 state.
So on the material synthesis and how we characterize them. So for our material syntheses, they're fairly simple. Basically, we're just taking our silver nitrate and molybdenum oxide, and then we combine them together with varying pH, and we get our battery material. So this is all true except for triclinic. For triclinic, we need to heat up monoclinic to 450 degrees Celsius for 5 hours. And here's a picture of all of our materials. As you can see, they're all varying slightly in color, so this helps us determine whether we're getting our product or not. But to actually characterize it and ensure that they are pure, we would use something such as X-ray diffraction and X-ray fluorescence. But those two aren't the focus of today. The focus is on scanning electron microscopy, or SEM. In SEM, we're getting these high magnified images, and these images are at a 5 micron scale, and SEM helps us to confirm that the structures are all varying and different. So for our monomolybdate, we have these blob-like shapes. Our trimolybdate, we have these bundles of nanowires. Our monoclinic, they are just plain nanowires, and our triclinic, they are once again these blob-like shapes. But if you remember, triclinic is made from monoclinic, so we have a theory that the monoclinic nanowires, when they're being heated up, they're creating these blob-like shapes. So for building the battery, first we need to make the cathode. So for the cathode, we take some carbon, our starting material, and binders combined together. We get this black slurry. Then we'll take our vacuum plate and spread a piece of foil on there, and then spread our slurry on top, and then use this doctor blade to spread it downward. Then we will let it dry, punch it out, and then we will take up our and press it and press the cathode to the foil. So to actually build the battery, we have a bottom connector, we have our cathode next, then we put some zinc sulfate solution on top of that. We put our separator, which is a filter paper, on top of that. Then we have our anode, which is a zinc metal piece, and then we have a bunch of other connectors, cap it, and this allows the electrons to flow completely through the system. And this is what it looks like when it's ready for testing.
So now we're going to talk about some of the results that we've gotten. So here is a discharge curve of our 4 battery materials. So if you remember from the beginning of the talk, Julie said that we want batteries with high energy and high capacity. So the more up and to the right the plot is, the better the battery is. So if you look here, our monoclinic sample performed the best with the capacity of about 250 milliamp hours per gram. Our monomolybdate and trimolybdate, materials performed about the same at 160 milliamp hours per gram. And our triclinic material performed the worst at about 100 milliamp hours per gram. So I'm sure that these numbers don't really mean a whole lot to anybody. So to give you an idea, the lithium cell phone battery has a capacity of about 300 milliamp hours per gram. So that means that our monoclinic battery could theoretically last almost as long as the cell phone battery. Dr. Smith actually made this awesome plot. So in black dots are zinc batteries that have been published as of 2018. And the blue dots are our batteries. So as you can see at these lower discharge rates, our batteries perform pretty well with everything else. But as we get to these higher discharge rates, our batteries start to become a lot more unique, which is good. Next, I'm going to show you some elemental mapping. So all of these images were taken at Argonne National Lab. So thanks to them for letting us use their instruments. So here on the left, we're starting with an SEM image of our monoclinic sample. And we can use the SEM to identify each individual element. So here in gray, we have silver. Here in molybdenum, here in blue, we have molybdenum. And here in red, we have zinc. And if you notice where we have these high concentrations of molybdenum and zinc, there's not really a whole lot of silver. So that tells us that this mechanism we proposed to you earlier of how our battery operates is actually doing this, which is good. So some of our future work that we want to do, we want to use an instrument at Argonne called the ACAT-TEM. It's one of 3 microscopes in the world, which is super cool. And this allows us to generate even higher power, higher magnified images. From the SEM, we got to the 5 micron scale, but hopefully with TEM, we can get to a nanometer scale, which would be super cool. And we are also testing rechargeability on our batteries. We've tried this before and it hasn't really worked out. So we're gonna try to add polymers to our materials to see if that will help with anything. We wanna thank Dr. Smith for letting us work with him. We wanna thank Brendan, Derek, and Jack for also working on the project. The BU Department of Chemistry for letting us use all their fancy gizmos here. EPIC for supporting undergraduate research. Indiana Academy of Science for funding. And Chicago State and Argonne National Labs for letting us use their instruments. Here's our references. Any questions?
Excellent presentation. The first question comes from Dr. Goins' research students who ask, do you personally have a favorite material for your batteries yet?
I do. I like monoclinic the most because it works the best.
Fair enough. The next question is from Paige. What are some common problems you deal with when making your battery? Are there any byproducts that are produced as a result?
So we did have some purity issues in the beginning, but we were able to get those problems solved. Like for example, when we were making monoclinic, we were having issues where we were also forming some triclinic, and that was mainly just a temperature problem that we fixed pretty easily.
Mm-hmm.
I don't know if you want to add anything.
No, but other than the monoclinic slightly forming a little bit of triclinic, I don't think we really had many issues.
All right, it looks like Laura had a similar question to Paige, so I'll move on to the next one from Professor Zygmunt, who asks, did the two of you do all of the steps in the battery fabrication process? It sounds like it takes a lot of time.
Could you repeat the question? Sorry.
Oh, yeah, he asked, did you two do all of the steps in the fabrication process?
So like creating the materials and assembling the battery?
Yes.
Yeah. So the synthesis typically for our monomolybdate and monoclinic samples takes about 5 hours. And if you look at our synthesis, mainly we just mix two things together and let it heat up. So if you know how to mix things, you can replace us at this point.
The trimolybdate was a little bit more difficult. We haven't really done much work on it because Brendan has thankfully left us some material that we can still be using. But other than that, the other three materials were very easy. And we also do build the battery ourselves.
Yeah.
Basically, it's just a series of punching out little holes and placing them on top of each other.
Yeah. A lot of trial and error, especially for getting nice coatings for our stuff.
Another question is what is the electrolyte you used?
So the electrolyte that we use is zinc sulfate. We want to use zinc ions, or we're using something with zinc in it because we are planning on oxidizing zinc. And we use sulfate because sulfate shouldn't react with anything for the most part.
All right. The next question is, how much of the performance of the battery is due to the monoclinic structure?
So we have two different discharge tests that we've done. This is a discharge with just adding carbon and binders to it. That's pretty typical in battery research because it helps you move electrons faster. And like builds like bridges, I guess, to allow the electrons to move. We have done pure pellet tests, and from those, our trimolybdate typically is the best. And we think that has to do with the fact that all of these extra molybdenum atoms can accept a lot more electrons. And we've also been working on finding like surface electron movement versus like diffusive electron movement. And we found that monoclinic typically does a lot more surface movement compared to the other three structures. So having like these nanowire structures, we think helps it take electrons better.
All right, great. And last question, how is ACAT-TEM different from other forms of TEM? Is the ACAT part of the microscope giving important new information, or are you really just looking to resolve images at a smaller scale?
That's more of a Dr. Smith question.
Yeah, we haven't really talked about it much. We just know that it's a very high-powered magnified
Microscope.
instrument.
Yeah.
So we just really want to get down to the nanoscale, but we're not exactly sure.
All right, Professor Smith says at Professor Venstrom, these samples decompose in TEM beams. The ACAT is a special low-dose electron imaging. Should let us distinguish silver metal from silver metal we form versus silver metal formed from beam decomposition. All right.
I believe it.
All right, thank you, Alan and Julie, for your talk. And I think we should move on to our last talk of the day, which will be by Trupti and Abigail. And correct me if I said your name wrong.
Yeah, it's Trupti.
Trupti, great. And they will be talking about, they'll be talking about understanding the role of RBFOX RNA binding proteins in brain development, neuronal function, and behavior.
Sorry, give me just a second. Oh my God, it's telling me to quit the meeting.
While they're setting up, I can just review the announcements for today, if that's okay. So just a quick reminder that the deadline for abstracts is Monday, July 13th, so this coming Monday. The deadline for uploading your poster is July 20th, and you'll get instructions from the Research Matters email account on Thursday about how to do these things.
Okay, can everybody see my screen now?
Yes.
Hello everyone, my name is Trupti and I'm gonna present on understanding the role of RB-Fox RNA binding proteins in brain development, neuronal function, and behavior. I'm going to start out by talking about why zebrafish are a model organism for our research project. Zebrafish are able to grow and mature at a very fast rate, making research very efficient. Zebrafish are vertebrates, which means that they have similar neuroanatomical features as mammals. Zebrafish eggs are fertilized and developed outside the body, and the eggs are transparent, allowing researchers to easily observe the developmental stages. The figure on the right shows the developmental stages of a zebrafish up to approximately 3 months. Zebrafish are capable of regeneration, including central nervous system tissues, which can help researchers understand the process of regeneration in the future. In addition, zebrafish are cheaper to maintain than mice, and they have more proliferative and neural regenerative zones, both of which are present in the cerebellum. The first part of our research project consists of understanding the role of RBFOX1L in brain development and behavior. RBFOX proteins are RNA binding proteins and alternative splicing factors that play a role in the alternative splicing of neuronal transcripts. Mutations of the RBFOX1 gene in humans have been implicated in the regulation of neurological and behavioral disorders, including autism, anxiety, epilepsy, and schizophrenia. Studies have shown that RBFOX1 knockout mice have seizures and increased neuronal excitability, as shown in figure C right here. In figure A on the right, it compares wild-type and RBFOX1 knockout mice using CFOS labeling, which indicates recently active neurons. The figure shows that RBFOX1 knockout mice have much higher CFOS expression in the amygdala and hippocampus regions of the brain when compared to wild-type mice. Then I'm going to also talk about the expression of RBFOX1L, which is RBFOX1L-like, and it has been identified in a restricted population of neurons within the dorsal telencephalon in the adult zebrafish brain. And this can be seen in this figure where parvalbumin 7 staining. It shows that RBFOX1L expressing neurons span DM, which is the amygdala-like, and DC, which is the putative cortical-like region within the zebrafish brain. As far as RBFOX1L, we are interested in understanding when RBFOX1L is first expressed in the zebrafish telencephalon during development. Preliminary studies have shown that RBFOX1L is not expressed in the brain at 3 days post-fertilization, but it is expressed in the adult zebrafish brain. We can determine the developmental time point when RBFOX1L is first expressed in the zebrafish by analyzing RBFOX1L expression at different stages of development leading up to adulthood. During the summer, we are focusing on analyzing embryos and larval stages. Once we know when RBFOX1L is first expressed in the zebrafish brain, we can determine whether RBFOX1L is necessary for neuronal development or function of behavior. And this can be determined by comparing mutant and wild-type zebrafish histologically, or we can observe the behavior of zebrafish at the time of when RBFOX is first expressed. The second part of this research project consists of understanding the role of RBFOX2 in cerebellar development and adult regeneration. RBFOX2 is the earliest RBFOX protein expressed in the brain and muscle in early development and may play a role in brain development. Specific deletions of RBFOX2 disrupt neural development and function, and such disruptions can cause anatomical changes. Within the brain, as shown in the RBFOX2 knockout mice, when compared to wild type mice, RBFOX2 knockout mice have a smaller cerebellum as shown in the figure. In adult mice, RBFOX2 is expressed in the Purkinje cell layer, but not in the granule cell layer. And as shown in this parvalbumin 7 staining figure, in adult zebrafish, RBFOX2 is expressed in Purkinje cells and also in the granule cell layer, indicating that expression in mice and zebrafish is different in the adult granule cell layer. So we are interested in comparing and contrasting the expression of RBFOX2 in mice and zebrafish, as well as determining whether RBFOX2 function will be similar in the zebrafish cerebellum or different, and whether RBFOX2 is necessary for regeneration of the cerebellum. And like RBFOX1L project, we are interested in determining the expression of RBFOX2 in the cerebellum during zebrafish development. For RBFOX2, preliminary studies have shown that RBFOX2 is expressed in the zebrafish brain at 3 days post-fertilization. During the summer, we will be focusing on analyzing RBFOX2 expression in zebrafish embryos and larval stage, similarly to the RBFOX1 project. Once we know more about when RBFOX2 is expressed in the zebrafish cerebellum, we can determine whether changes in cerebellar development will occur in RBFOX2 mutant zebrafish and whether RBFOX2 is required for regeneration of the cerebellum. We can assess neural production in the adult cerebellum after injury by conducting an EDU pulse chase, as shown on the right over here. An example of this would be doing a needle stick injury and then labeling the cells with EDU. With EDU would label the cells that are in S phase, including progenitor cells. And after a period of time, which is known as the chase period, we can determine the number of newly produced neurons by quantifying the number of neurons that are EDU positive. And this figure shows how the EDU pulse chase would have a combination of the cells. I would like to thank Dua and Romeo, who have also worked on this project in the past, and Dr. Michael Berberoglu, who is my advisor, Valparaiso University Biology Department, Indiana Academy of Science, and the University of Guild for undergraduate research. Thank you.
All right, thank you. Good talk. All right, let's see if we have any questions in the chat here for Trupti. Question from Carter. Do you have names for the fish?
No, we don't have names for the fish.
Carter would totally name the fish. Question from Laura F. What is a knockout mouse?
Knockout mice would not have the RBFOX gene for example, for this one, the wild type would have the RBFOX2 expression, and the ones that are knocked out would not have the RBFOX expression. I don't know if that made sense.
That makes sense. And Laura says, okay, thanks. And what does that help tell you when you have one with and one without?
Okay, so for wild type, if they have it, then we can see what the difference is if there was no RBFOX expression. So we can understand what kind of role it plays in a brain development maybe.
All right, good answer. I'm just trying to decipher the chat. And following up, there's another question before this, but I want to just follow up. Maggie asks, so RBFOX occurs naturally in the wild types?
Yeah.
Okay. And then another question, how long does it take to get results since you have to wait for them to develop?
So right now we already have some adult zebrafish, but when we do crosses, it takes up to 3 months for them to fully develop to a adult zebrafish. So like this, they grow pretty fast.
Wow. Yeah, so they grow pretty fast, but 3 months, like that's a pretty big commitment.
So while we're doing research, we keep doing crosses in between.
And also I can add something there, just keeping in mind too that the larval experiments can be done within 2 or 3 days because the zebrafish develop quickly during development.
Oh, great. So one more question, and then since we're running over on time a little bit, we'll wrap up. The next question is from Peyton, who asks, what would be some of the applications for this? I imagine there's a lot.
So once we understand what kind of roles RBFox1L and RBFox2 play, I'm more familiar with the RBFox1L project. So for that one, we can do behavioral studies. So we can use them to, like there's an assay called novel tank assay where we can transfer the zebrafish to a new tank to assess their anxiety levels, which can also correlate to humans, because we have the RBFox1 gene.
All right, excellent. Thank you, Trupti, for your presentation today, and thank you to all of our presenters. We'll see you next week, and don't forget your abstracts.
I thought we had really good questions today. So.
Sorry, I was talking with my microphone off.
Oh,
Yeah,
that's okay.
it really helped to sort of lower the bar for like what you have to ask, because it can be like intimidating to think like, oh, I have to ask about their data or something really specific about their methods.
Yeah, and the group, the advisory committee or the steering committee, speculated that last week we heard more of the mathematical talks, and I think that for some people that's pretty intimidating.
Yeah, I can see that. Yeah, that was a great.
Well done. Yeah.
All right.
Sorry, I cut you off. What would you say?
Oh no, I was just gonna say. Is there anything else that I?
No, I just thought it was really great, and it's gonna run a little bit over with 4 talks, so I actually think that people kind of enjoy this because it's one of the few times where they get to connect with a wider group.
Yeah, that's true.
Yeah, so 5 or 10 minutes over, I don't think is a big problem at all.
Okay, if we're still going at 2 next week, then I'll.
Yeah, at some point we do need to cut it off. Yeah. Good. And I think I didn't time them all, but it seemed like the